It's time to start. Good afternoon, everyone, and welcome to the ESDR Kitchen. I am Jeremy Domizio, and today I'm co-chairing this session with Marianne Lövendorf. And today it's a freshly baked episode that is nicely sponsored by Takeda. It will feature two recently published articles that made a high impact in dermatological research. The first presentation will be done by Anahi Odell. We'll talk about skin fibrosis, a new pathway in skin fibrosis. And the second speaker is Gabriela Raimondo, who will talk about psoriasis and how the inflammatory response is spread from the skin to the joints. So now I will let my co-chair introducing the first speaker.
Yes, hello everyone. So I will introduce Anahi Odell, who is an associate research scientist in the Department of Dermatology at Yale University. Anahi earned her PhD in microbiology and molecular genetics from the University of Vermont and began her research studying toxoplasmic gondii with a focus on host pathogen interaction and drug target identifications. Her current research focuses on the molecular mechanism driving fibrotic skin disease, and her work investigates how epidermal growth factor receptor signaling regulate fibroblast activation and contribute to fibrosis in conditions such as scleroderma. And this is also something that we will hear in her talk today. So, Anahi, please go ahead. We're looking forward to this presentation.
Thank you, Marianne. I'm going to start sharing my slides. Okay. So I'm going to tell you about a recent work that was published at the end of last year on the role of EGFR STAT1 pathway driving fibrosis initiation in fibroinflammatory skin diseases. So we started out this project with the question of what really makes inflammatory skin diseases fibrotic or non-fibrotic. So in order to answer this question, we integrated publicly available data sets of atopic dermatitis and psoriasis for examples of non-fibrotic skin diseases, and systemic sclerosis, graft-versus-host disease, lupus, and hidradenitis suppurativa for examples of inflammatory skin diseases that feature fibrosis. And then we wanted to look at what is different than what is expressed in the SFRP2 positive fibroblasts, which are the precursors for myofibroblasts. And when looking at the differentially expressed genes, in particular in the fibrotic skin diseases, we were able to identify a very interesting description of the niche in the skin for each disease. For example, for hidradenitis suppurativa, we found that it really features cytokines that promote neutrophil activation, and it really correlates well with the inflammatory conditions in the skin. In the case of lupus, it's very much driven by interferons. And in the case of scleroderma, for example, we see features of growth factor signaling, a very oxidative stress environment, and mechanosensing. And then about the differentially expressed genes that are shared among these fibrotic skin diseases, one that really stood out to us was STAT1, which is a non-transcription factor to be associated with interferon signaling downstream of JAK kinases. So we wanted to explore this a little bit further. So one thing that we wanted to ask was, does this increase in expression of STAT1 correlated with increased activity? So in order to understand that, we did CINEC analysis, which takes into account the expression of that transcription factor, along with the putative transcription factor binding sites in all the genes in the genome, and the expression level of those genes. And then it defines a regulon that we can then analyze how enriched it is in the different data sets. And interestingly, STAT1 was the most, or one of the highest enriched regulons in all the fibroinflammatory skin diseases. And this analysis also pointed to regulons enriched that are related to growth factor signaling, like EGR1 or JUN. So we really have this idea that EGFR and STAT1 signaling, there might be a crosstalk. So we wanted to see if this happens in vivo and if this is relevant to fibrosis. So one of the first experiments we did was a bone marrow transplant, where we essentially asked the question, are immune cells that are radiosensitive, STAT1 in those cells is still required for fibrosis? And we found that it is dispensable for fibrosis development. So we had this idea that perhaps it truly is STAT1 in the fibroblast. So in order to address that question directly, we created STAT1 conditional knockouts in fibroblasts, and then tested these mice in a bleomycin mouse model of skin fibrosis. And these mice were protected from fibrosis. And interestingly, this phenotype could be phenocopied by treatment with an EGFR inhibitor, gefetinib, showing that there truly is a crosstalk between the two signaling pathways. So we wanted to see if this really happened in fibroblasts, and it's been known in the cancer field that EGFR can directly phosphorylate STAT1, even though up to now it really wasn't clear what would be the consequence of that phosphorylation event. So first we wanted to see in human fibroblasts, does this actually happen? So we treated human dermal fibroblasts with the whole array of EGFR ligands. And we found that the EGFR ligands that have high affinity for EGFR are the ones that show increased phosphorylation of STAT1. But all the ligands in our hands were able to activate downstream signaling pathways from EGFR, like AKT and ERK. Now, out of those ligands, especially the high affinity EGFR ligands, which one of those are actually expressed in high levels in the skin? And the one that really stood out was TGF-alpha, and it's expressed by keratinocytes. Now, which keratinocytes are the sources for TGF-alpha? It's known from a long time in the field that activated keratinocytes are mainly the source of TGF-alpha in the skin. And so we wanted to see, are there activated keratinocytes in fibroinflammatory skin diseases? We've known for a while that activated keratinocytes are present in atopic dermatitis and psoriasis. And now we found that activated keratinocytes are also, it's also a phenotype of keratinocytes in fibrotic skin diseases. And we were able to validate this result by immunofluorescence of skin. In healthy, you really see only expression of the stress keratin 16 in a very limited area in the epidermis, and it doesn't really overlap with expression of TGF-alpha, whereas the expression of keratin 16 is much increased in fibrotic skin diseases, and it overlaps perfectly with major cells expressing TGF-alpha. Now, at the time that we published this paper, our single cell data set was not strong enough to detect the significant increase of TGF-alpha expression in the activated keratinocytes. But we have since improved our data set by adding more diseases onto our atlas, and that gave us better granularity on the analysis. And we've been able to find that TGF-alpha is indeed significantly increased in expression in activated keratinocytes. So now we have this dichotomy and this question of, okay, so EGFR leads to increased STAT1, and it's been known for a long time that interferon leads to increased STAT1 through a completely different pathway. So we wanted to see, does EGFR activation of STAT1 require JAK kinases? So we treated dermal fibroblasts with interferon and TGF-alpha. And we pre-treated these cells with either JAK inhibitors, ruxolitinib, and deucravacitinib. And separately or combined, we were able to completely abrogate STAT1 phosphorylation in response to the interferon, but it does not affect STAT1 phosphorylation in response to TGF-alpha. On the other hand, treatment of these cells with an EGFR inhibitor, gefetinib, does lead to a decrease in phosphorylated STAT1. So what would be the consequence now of activating STAT1 from two separate pathways? So what we did to answer this question was use fibroblasts isolated from STAT1 knockout mice, and then treated these fibroblasts with TGF-alpha. And we found, as expected, that interferon-stimulated genes are dependent on STAT1, but are not responsive to TGF-alpha. However, there's a subset of genes, and those are fibrotic genes, that are both responsive to TGF-alpha and dependent on STAT1, really defining a profibrotic profile that requires STAT1 and is responsive to growth factor signaling. And this result was we were able to validate it pharmacologically using a STAT1 inhibitor, fludarabine, that has been described for a long time as a chemotherapy. But it also is capable of binding the SH2 domain of STAT1 and thus inhibits STAT1. So we used this inhibitor at non-cytotoxic levels, and we were able to validate that inhibiting STAT1 abrogates the TGF-alpha induction of fibrotic genes. So this leads us to a proposed model where keratinocytes, which are activated in fibrotic skin diseases, produce and secrete TGF-alpha to then act on the fibroblasts, activate EGFR, and through activating STAT1, promote the transcription of a pro-fibrotic transcriptional profile. And with that, I would like to acknowledge everybody in the Odell Lab, our funding sources, and thank you for the ESDR Kitchen for allowing me to present our work.
Thank you very much, Anahi. That was a really good presentation. I forgot to tell the audience that if you have any question, please use the chat box and send us your question. We'll do the Q&A session at the end of this episode, so after the second talk. So now let's move to the second speaker, Gabriela Raimondo. She earned a medical degree at the University of Milan and then completed a residency in rheumatology at HPN Hospital in Milan. During her training, she was actively involved in translational research on autoimmune diseases and collaborated with University College Hospital in London as a clinical research fellow. And following her specialization, she joined the university clinic in Erlangen, Germany, where she enrolled in a clinical scientific program that allowed her to integrate basic research with clinical practice. And she later took on responsibility for the biopsy unit within the Department of Rheumatology at the University Clinic Erlangen and became a young PI. So today she will present her last work published in Nature Immunology, dealing with myeloid precursors in the skin and fibroblasts in the joints and how they spread the psoriatic disease from the skin to the joints. So Marianne, please, the stage is yours.
Thank you, Jeremy. I'm going to share my screen. So thanks again also for having me here today. It's a pleasure to present our recent publication. We all know that around 30% of patients with psoriasis will develop over time joint manifestation, and these events will actually lead at some point to the diagnosis of psoriatic arthritis. However, what we don't know is why this is happening and why some patients experience over time a kind of migration of inflammation that most of the time starts in the skin and then lands to the joints, whereas other patients don't. In order to answer this question, we hypothesize that a skin joint axis might actually exist and might explain the psoriatic arthritis pathogenesis. This was our hypothesis, and in order to answer and to investigate this hypothesis, we needed to experimentally reproduce in vivo what we observe in our clinical practice. And we managed to do this through the systemic overexpression of IL-23 in Balcin Black 6 mice, because this will enable us to generate two different mouse models. In case of Balcin mice, the IL-23 was able to induce skin and joint manifestation resembling psoriatic arthritis, whereas in the case of Black 6 mice, the IL-23 was able to induce only skin inflammation resembling then psoriasis. But we still needed to address whether there is a crosstalk between skin and joint. And in order to do that, we actually use specific transgenic mice, which are called Takeda mice. These we had in Balcin Black 6 background. We overexpress here also IL-23, and we use these mice because all cells in these mice express the Takeda protein, which is a fluorescent protein with a green fluorescence. So when we expose the psoriatic skin lesion to a UV light source, we are able to induce a photo switch from green to red. This is very handy because all the Takeda red cells that originate from the skin then can be targeted and follow, for example, towards the joint. And here, for the first time, we confirmed that there is indeed a skin joint axis. So there are migrated skin cells landing into the joints already on a steady state. However, the IL-23 was able to amplify this migration event. We also confirmed, or at least we actually proved, that most of the cells migrating in the joint are myeloid cells, so CD11b positive cells. However, we did not see any difference in terms of frequency between psoriasis and psoriatic arthritis mice, suggesting that this is a psoriasis-related event. We then sorted these cells and we performed single-cell sequencing. Again, we confirmed that most of the Takeda red cells, so the cells coming from the skin, were dislocated. We did clusters of myeloid cells. We performed different bioinformatic approach to actually identify the original myeloid precursors landing in the joint and then differentiated towards macrophages as cells expressing markers like CD2 and MAC class 2, and also have an upregulation of genes related to immune cell migration. But can we actually identify these cells also in human? Well, the answer is yes. We integrated our single-cell sequencing dataset from the mice with human single-cell sequencing dataset of psoriatic arthritis synovium, and we could identify our myeloid precursor population, and we could also see them in the single-cell sequencing dataset of psoriatic skin lesion. But can we actually confirm the migration hypothesis also in human? In order to answer this question, we performed paired biopsy of skin and synovial tissue of patients with psoriatic arthritis with an early diagnosis, and we perform mitochondrial DNA sequencing. So why we use this approach? Well, it is because it is known that cells sharing the same somatic mutation of the mitochondrial DNA belong to the same lineage, so they have the same origin. And indeed, we showed that the myeloid cells, followed by T cells in the synovium, share the highest amount of somatic mutation with the skin. And within the myeloid cells, it was indeed our CD2 positive myeloid cells, the myeloid precursors, sharing the highest amount of somatic mutation with the skin, suggesting the skin origin of these cells. But then when we back to the mice and we quantified the amount of these cells, again, there was no significant difference in terms of frequency between psoriasis and psoriatic arthritis mice. So are these cells relevant at all? Well, the answer is yes, they are, because when we adoptively transfer these cells in the psoriatic arthritis mice on an early time point, we are able to significantly aggravate the inflammation in these mice on an early time point, as I said. So this means that these cells are necessary, but they are not sufficient to initiate the inflammatory process. So what is here the second hit? To answer this question, we look at the transcription profile of these cells at the time when they enter in the joint in psoriatic and psoriatic arthritis mice. And we saw that at the beginning, they are very similar. But as soon as they start to differentiate towards macrophages, then they start to differ. And the macrophages in the psoriatic arthritis mice, they have a pro-inflammatory phenotype, whereas the macrophages in the psoriasis mice, they have an anti-inflammatory phenotype. Meaning that something is happening in the joint that can actually influence the destiny of these cells towards a more pro-inflammatory or anti-inflammatory phenotype. And this something is actually the fibroblast. Especially, we saw that in the psoriasis mice, there was a very strong interaction between the skin-derived cells landing in the joint and the fibroblast, in particular the CD200 positive fibroblasts, which are known to be pro-resolving and anti-inflammatory. Indeed, when we quantified in the joint of our mice these fibroblasts, we saw that the mice who had psoriatic arthritis, so joint inflammation, had a significantly drop down of the CD200 positive content, whereas the CD200 positive fibroblasts stay stable in the mice who are protected from joint manifestation. Very importantly, we also observed that our myeloid precursors highly express the CD200 receptor. So is this CD200 receptor, CD200 axis relevant? Actually it is, because when we take our psoriasis mice and we inhibit the CD200, then here we are able to initiate the inflammatory process in the joint. We even confirmed this in an independent psoriasis model, which is based on the local application of imiquimod. Again, here we first validated that we could see an enrichment of migratory myeloid precursors, so the CD2 positive myeloid cells in the joint treated with imiquimod, again suggesting that this is a psoriasis-related event. However, these mice do not spontaneously experience arthritis, and indeed the CD200 content of the fibroblasts in the joint is actually stable. But as soon as we inhibit the CD200, then we are again able to initiate the inflammatory process. What about human? Here we perform imaging mice cytometry approach on synovial biopsy collected from psoriasis and early psoriatic arthritis patients. And we observed that the CD200 content was highly enriched in the psoriasis synovium. And again, that in this tissue, the psoriasis tissue, there was a highest interaction between the CD200 fibroblast and the CD2 positive myeloid precursors. We also further investigated the downstream effect of these CD2 positive myeloid cells, and we observed that in the psoriatic arthritis synovium, there was actually a lower distance between the CD2 positive myeloid cells and the CD8 positive T cells, which are known to be very important in psoriatic arthritis pathogenesis. And again, we also proved that when we do co-culture of the T cells together with the CD2 positive myeloid precursor, then we are able to significantly increase the release of IL-17A. So in conclusion, we are here suggesting a two-hit hypothesis. From one hand, we need the migratory influx of myeloid precursors coming from the skin, landing into the joint, but then we need the microenvironment. And in particular, it is very important the CD200 positive content of this joint, because they play as a gatekeeper in defining who is gonna switch and develop arthritis and who is not. With this, I'm very happy to conclude, and thanks all the people that participated in this very huge work, and I'm very happy to answer all your questions. Thank you.
Thank you very much, Gabriela. And now the session is open to questions from the audience. I remind people to send their question in the chat box. I may start with a question to Anahi first. So do you think that the pathway that you're describing is specific to the skin, or is it specific to fibrosis in general? So it could occur in the lungs, for example.
I do think it's specific to fibrosis. As far as whether or not it's specific to the skin, that's a great question. I do not know. What I can say is that even though our gut feeling tells us STAT1 is relevant in immune cells, we know JAK inhibitors improve fibrosis. It's been shown by others and by us. But really, that aspect of STAT1 as a transcription factor relevant for transcription of fibrotic genes in a mesenchymal cell type, it really is completely JAK independent. And we're trying to shed light in there's more to STAT1 than JAK inhibitors. And now, could it play a role in fibrosis in other organs? It's possible. We have not looked, and that would be very interesting to look into that.
Okay, I have a short question as well for you, Anahi. So as far as I recall, the STAT1 expression was also a little bit elevated in psoriasis.
Yes, it is.
So what do you think about this? Because what is the function then for this disease?
Exactly. The expression was a little bit elevated, but the regulon in the CINIC analysis was not enriched. So, so we're thinking, you know, I really don't know what to think, to tell you the truth. The expression itself is increased, but all the regulon, the genes that it's regulating, they're not showing strong STAT1 activity as a transcription factor in psoriatic skin fibroblasts. So I wouldn't really draw too many conclusions from that.
Okay. So another question for you: did you try, or do you have plans about, isolating fibroblasts from some of these skin diseases, so you can work with those, like for in vitro assays or something like that?
We do. We routinely do that, and then compare, mainly from scleroderma patients. the last author in the publication, he's a dermatologist, so he has direct access, but he does a lot of mostly translational work where we work with the patients and you compare, their fibroblasts, and we can isolate keratinocytes, and there's a lot of in vitro work that we can do thanks to being able to have a direct connection with the patients, and that's been really great.
Yeah, that's great.
One more question, maybe to Anahi,
Yes.
to finish on that topic. So some patients receive EGFR inhibitors. So do you know if these patients have somehow differences in skin fibrosis, if they develop fibrosis on the side, or?
The problem with the EGFR inhibitors is that they're very toxic. So it is really not the best option for treating fibrosis. So that's why a prior publication from the lab that Ian Odell led was, instead of trying to target directly EGFR, he found that there's a very strong, relevant interaction between EGFR and epiregulin, another one of the EGFR ligands. And instead of targeting EGFR, if you target epiregulin directly, then it improves fibrosis. Not just not prevent it, but improves fibrosis, which is really, that's the golden egg of trying to treat fibrosis, because the patients show up with fibrosis already established. And he's been able to show that by using a monoclonal antibody to target epiregulin, he's able to improve fibrosis outcomes. So I think that's a better option rather than trying to target EGFR directly.
Makes sense.
Thank you.
Okay, Maria, I have a question for you. Let me see in the chat. No. Okay, so there are a few patients that develops only psoriasis arthritis and not have skin involvement. So have you any thoughts about, your proposal of how it works, that it comes from the skin, because these patients would probably not have it in the skin?
Well, I mean, this is a very important and relevant question, of course, raised also from the reviewer. But we were prepared for that because, of course, this is a model that we are explaining where there is this combo skin and joint. We look at this experimentally where we can control, of course, the disease, and we could see that indeed there was first the skin, and then on a later stage there was the joint inflammation. And most of the patients actually reflect this combination. They have psoriasis for several years, and then at some point they develop the joint inflammation. Yeah. Of course, our model cannot explain all. We don't know, for example, if the patients who have a familiarity for psoriasis, for example, while they don't have skin involvement per se, if in this patient there is a higher migration of these CD2 positive myeloid cells, for example, towards the joints. This is something that with this model, we cannot, of course, completely answer. So we tried to have a mechanistic answer to those 70% of the patient that in the end have this pattern somehow, or more roundabout 70-80%. But of course, the patients who are without skin inflammation, this is something that we still need to work on and address. It would be interesting to see, for example, whether they have a higher migration of these myeloid cells or not, or the CD200 fibroblasts in the joint.
Thank you. Let me see, there is actually a question. Let me see.
To whom?
Yeah. Who is it for? Oh, it's for you, Maria. So Janis writes, when I read your publication, I stumbled across the fact that mitochondrial DNA was used for tracing. Is this leaked mitochondrial DNA upon inflammation?
So this technique was mostly used first also in oncology, for example, for metastasis. So we look at the SNPs, and you can actually identify specific feature and signature across the mitochondrial DNAs, and they are known to be more conserved. So that's why it is used as a lineage marker in a way to identify cluster of cells that have the same origin. So this is why we use it for the trafficking. Actually, it was a suggestion of the reviewer, indeed, and it works also brilliantly for us. So we were the first one suggesting this also in autoimmune disease. So I hope that this will will be used also further for trafficking, for example, not only in metastasis but also in inflammatory process.
Okay.
I have
Cool.
another
Thanks.
question to Gabriela. In patient, do you think there is any way to predict the patient that will develop psoriatic arthritis if we analyze these myeloid precursors in their skin?
So
And
a very good point. We also found these cells in the PBMCs of a patient with PSA and PSO. It was significantly increased compared to the healthy donors. And this is, I think, where we can work further. If these cells alone can really predict the development of psoriatic arthritis, this still need to be tested, but maybe we can use to at least identify the high risk patients. This is something that needs to be tested with clinical trials, probably. Yeah, but it would be good to go in this direction for sure.
That's really interesting. Yes, indeed.
Yes.
Marianne,
So,
I think we have to close. Yeah.
Yeah. So there are no more questions. So I think with that, I would like to start by thanking the two speakers for some wonderful and very exciting talks. And it's very extensive work and impressing. So thank you very much for that, both of you. And then I will just make a reminder about that all the ESDR Kitchen episodes can be found on this webcast, and they are freely available, and you can find them on YouTube, or there will also be a link on the ESDR homepage. And then a short reminder about the next ESDR Kitchen episode on May 20, and this is a collaboration between the ESDR and the JSID, and the theme is about genetic architecture on skin diseases. So please stay tuned for that episode. Finally, just a quick announcement of the annual meeting, the ESDR annual meeting in September in Heidelberg. There are the deadline is coming up next week, so I hope to see many of you there. There's already list of confirmed speakers, and the program is very interesting this year again. So I hope to see you there. And with that, we will close this session and then have a nice day.
Bye.
Bye.
Bye.